case, and thanks to speak again. And the next talk will also be from Israel, and Professor Per, please.
Excellent. Just this one? Okay, great. Okay, thank you. Our lab is studying how to manipulate cellular function in order to generate novel therapeutic approach to treat mainly leukocytes-implicated diseases. As you see, it's a huge field, and all those bluish diseases is some active work that we are doing. And actually, I'm going to highlight something that is only half related to leukocytes, but nowadays we know that immunotherapy is becoming more and more interesting, so maybe they're also a good candidates. So I want to highlight an example of systemic and localized treatments, basically using RNA molecules, and I'll talk about GBM and general strategy about T cells. So our lab is divided into two major groups, people that are doing drug delivery and protein engineering to create a more precise targeting approaches, and the other part is doing exome or transcriptome sequencing in order to find new drug targets, and of course the ideal situation will be to join forces in a kind of a personalized manner. The first part of my talk, I'm going to talk about glioma, and the person behind this work is a collaboration we do with the neurosurgery department in one of the biggest hospitals in Israel with Dr. Tzvi Cohen, and Srinivas Rameshetty is the guy behind this work, and there is also a poster, and he's also giving a short talk. So basically, glioma is a devastating glioblastoma multiforme is a devastating disease, and basically there are many, many challenges, mostly the infiltration of this tumor into the brain. It's very hard to stop it, and the median survival is about 14 months. And we looked at expression of some proteins, among them the CD44. And CD44, as you can see from staining of patient samples, it's quite high, and if you look at, doesn't really work well. Okay, and if you look at the expression of cell lines, also you can see quite high of an expression. Now, CD44 is a complex molecule. It has 10 alternative splice. Actually, recently, some people suggested that there are actually a few more splice variants, and there is a structural differences between those between the standard form, which is mostly expressed, I can say, everywhere, towards the variants. And the variants have a stem area that can tilt the molecule, and it will influence the binding as well. And we set a few papers over the years in targeting strategies using conformational sensitive approach, basically going after one conformation and not the other, and published some papers on this. But in this work, we use a microfluidic system that is here. Actually, you can see outside precision nanosystem. And if you're interested, they'll be happy to answer any questions. And the idea behind this microfluidics is basically use RNA on a slightly acidic buffer with lipids. And we're using ionizable lipids, so basically in an mildly acidic pH, they're positively charged, and in a neutral pH, they're almost neutral in charge, and at the end, you get very robust, very nice particles, so just remember this. And at the end, you can actually use the natural eigen, hyaluronic acid, and we have been working with this for the past almost 20 years in different permutations, and you can bind to the surface, and this is a short hyaluronic acid, about 5,000 molecular weight, and this is some characterization, so about 100 nanometer in diameter. They have like a flower, basically, conformation or look like this, and they bind very specifically to glioma in a spheroid culture, so they look very nicely from patient samples. And then you have to choose an appropriate target, and we use PLK1, pyrrolide kinase 1, and this is a G2M inhibitor in the cell cycle, and it's influencing lots of other genes, but at the end you can actually visualize and see cell cycle arrest and apoptosis at the end. And if you perform those experiments under shear flow condition, so basically only binding particles will be around, This is the amount of gene silencing that you can add ex vivo. Basically, it's about 80% knockdown. It translates very, very nicely into cell death over time. And then the problem is that you don't have a really great model for glioma. And one of the things is to use an orthotropic model. So using a stereotactic instrumentation, and this is just a simple illustration, you basically put 3 microliters of cells into the brain in a localized manner. So this, you get a tumor, and the tumor, again, is not the human tumor. So it's not infiltrating deep enough. And this is one of the problems. There are some genetic models that try to mimic them, but none of them are really similar to the human disease. And anyhow, it's a model, and now you have a model in hand, so you can do some Cy3 delivery of siRNA, or Cy3 labeled siRNA, in this U87mg models. And basically in time from 3 hours, 6 hours, and 24 hours, you see more and more staining of this area of the glia. And then if you look at therapeutic gene silencing, you basically, after a single administration, can take out the cells sorted. These are human cells with anti-CD44 variant 6, which is expressed on those cells, and you can get about 75% knockdown. If you look at survival rates here, and these are just the SI control particles and the SIPLK, we can prolong the survival dramatically after 4 administrations. So the administration are locally administered into the tumor bed area. So we're talking about an administration at day 7, 9, 20, and 22. We also believe that PLK1 reduce the blood vessels around the tumors. But this was published, and we are now moving to the next step, which is a scale-up process of this. The second story is a collaboration that we have with Peter Coulis from Vancouver, Alnylam Pharmaceutical, people at the Methodist Hospital Research Institute, and Derek Dextron from Miami School of Medicine. And here we tackle a problem of systemic gene silencing in primary T cells using targeted lipid nanoparticles. And this work was done in collaboration between 2 very talented postdocs, Srinivas Ramishetty and Ranit Kedmi, both presenting posters here, so I think you should go and talk to them. And why is the reason? Why do we want to target CD4 cells? So for us, it's a proof of concept from one side, but the other side, you can actually look at how many roles T cells, CD4 cells have. Okay, so of course they interact with antigen-presenting cells, and they can differentiate using different transcription factors into the Th1, Th2, Th17, Tregs, Th9, and THF, and others as well. And it's becoming more and more interesting that these are central cells that you want to manipulate their fates. So how we produce this, we again use the microfluidic system. And here we introduce also some malamide, and everything is published, so you can actually go and trace them in the papers. And then you introduce an antibody basically by reducing it. So after reduction, you need to purify this, and this is done by gel filtration column. And at the end, you can collect the anti-CD4 LNPs that have siRNAs inside, and you get a very high encapsulation in this, almost 100%. It's a very robust process, and then you can validate this, the specificity, the efficiency, etc. So I'm not going to go into details. We have 3 types of particles. Particles that have a targeted system, anti-CD4, and a specific clone. System that have an isotype control on the surface, and just regular particles without any antibodies. And just that you see that basically in terms of the geometric mean fluorescent, if we entrap siRNA inside that are fluorescent, or if we entrap the CD45, which has a labeling, basically we see that very specifically they will target CD4 cells. So the isotype control will not basically cause selective targeting ex vivo. Now, if you go and look at specificity, here you have CD4 positive, CD8 positive, and CD3 minus. It means everything but T cells. So it means B cells, macrophage, dendritic cell, neutrophils, etc. And you see how specific this is, and this is a delivery of Cy5 labeled siRNA. So it goes very specifically into those cells. But if you look at the amount of silencing that we get, you get actually a surprise. So they're all, and this is a surrogate marker. We use CD45 because we can trace it in flow cytometry. So 99% of the cells are T cells here that gets this siRNA. but the amount, the level of silencing is not high. It's about 20%. And now the question is, can you actually feature something biologically relevant with a 20% knockdown? And the answer naturally is no. So in the next step, we wanted to look at different organs, and these are all systemically administered particles. We wanted to look at different organs that have CD4 cells, okay, spleen, lymph node, bone marrow, blood, etc. And you can look at the level of silencing, and it's not really a lot. So the best one is about 35% knockdown. However, we also observe that we have two different populations. We have CD4 high cells and CD4 low cells. And then the hypothesis was that maybe we are now in the process of internalization. So let's assume now that the nanoparticle that have an antibody is now internalized, and if we come with another antibody that wants to see what's happened on the surface, it's not there anymore, okay? So maybe the ones that are CD4 low are in the process of internalization. Can we correlate this to silencing levels? And is it going to be improved? So what we have done is quite a simple experiment. It's just do a sorting. Okay, so you inject the nanoparticles IV, and then you collect splenocyte after 1.5 hours. We actually look at different time points, but already it happens 1.5 hours. And then you sort the cells to two populations, CD4 high and CD4 low. Okay? And then you can actually visualize, and there is a lot of controls here in terms of coming with an anti-rat FC that goes under your particles, or you can go and directly see it, visualize it with sRNA against Cy5. But basically you have distinct two populations, CD4 high, CD4 low, and it's very nicely correlated to the silencing effect. So if we took the targeted system that is CD4 high, the amount, and this is just a representative example, is about 16% of the cells are silenced. And the amount of silencing, sorry. If you take CD4 low, the amount of silencing is about 70%. And if we create an average, you will find out that these guys can silence about 60%. Okay, so the CD4 lows are the ones that being silenced. The CD4 high are minimally silenced, and the isotype control is almost irrelevant here. And this is also my take-home message here. So there are two things that needs to be taken into consideration. One is how fast is your systemic target being internalized? There is always an agreement or disagreement about targeting approach. Should we target or not target? Usually I call it to target or not to target, but basically, if you look at circulating cells, there is another barrier. If you look at lymphocytes, about 80–85% of the lymphocytes is a nucleus. For creating RNAi work, it has to be really internalized fast. And we have shown in the past, in a sets of paper, that LFA1, for example, is a great target because the recycling process is 45 seconds. But only one of the antibody out of 18 against LFA1 can do it. It has to be a ligand binding domain antibody, a blocking antibody. In this case, in the CD4, we know also that it's internalizing. Okay? How fast it's in the internalization is very, very important. So this is one point. And the other point, I'm finishing. The other point is that we have to be extra careful about the immune response. So one of the things that could happen also is elimination of the particles, and that the amount that will reach the target is very, very small. So what is the state? Why we do get two different populations? Is it activation? Is something else is happening? Is it the siRNA, sorry? Is it the lipids, the antibody, the combination? Of course, if we drop the FC portion, something else will happen. But this is the take-home. So be careful and choose your targeting moiety based on the biology, not based on the fact that you have an antibody available for use, and also calibrate it, because if you have too many antibodies, it might not work for you. And if you have antibodies that are not binding in the right conformation, you're actually going to get a better targeting with a non-targeted system. So at the end, I just want to thank the people in the lab. And again, if you want to hear more, you should go to the poster and to the short talk of Srinivas and Ranit, who is here, and the funding agency, and thank you so much for listening.
Thank you very much. We have time for one question. Please, use the mic.
Great work, Dan. Just a quick question about your GBM model that you're testing these. How does it get across the blood-brain barrier?
It's not.
So how are you injecting?
The particles are injected intratumoral.
Okay.
So basically, you can have a 1 Tesla MRI, just T1 to see that they have a tumor there, and you inject into the tumor bed.
Can it work also for pancreas?
Locally? Maybe. We have not injected. I can tell you that in a different system with hyaluronan, if you inject systemically, you get penetration into the pancreas. But it depends on the size of the tumor and the diffusion rate, which is a very challenging situation in solid tumors.
So thank you very much again. And we come to the next.